The bacterial flagellum was one of the central examples of irreducible complexity Behe highlighted in his book.
If you take away the propeller, if you take away the motor, if you take away the clamps that hold it onto the cell's membrane, take away any of a number of different parts, it's not that the flagellum is going to spin half as fast as it used to, or it's broken. It doesn't work at all. It's like taking the propeller off of an outboard motor on your boat and wondering how far now you can go in the water. You can't go anywhere. So that's a problem for Darwin's theory, because Darwin's theory says that things evolve by working a little bit, maybe not very well, but a little bit, and then a mutation, a change comes along that helps it work a little bit better, and that helps the organism survive and have more offspring, and so then another change comes along, and another, and it gradually builds up to the final structure. Well, that might work for some things, but it doesn't work for systems that are irreducibly complex, things like the bacterial flagellum. Because, well, if you wanted to build an outboard motor for a boat, what would you start with? Would you start with, say, just an iron rod that, in the future would attach a motor to the propeller? Well, what's that going to do? It's not going to do anything. Would you start with just the propeller? Well, that's not going to do anything. It's not attached to anything. Would you start with just the motor? Well, that's not going to propel you anywhere. So with irreducibly complex systems like the flagellum, Darwin's idea is dead in the water, like a boat with an outboard motor that doesn't work.
Natural selection selects or favors in variations that confer a functional advantage on a system. Many of the simpler versions that you could imagine of the bacterial flagellar motor perform no function at all. So if you imagine trying to build a flagellar motor, adding parts one by one until you finally get to the complete system, you're going to encounter configurations of parts that confer no function, in which the motor simply will not work, at which point the evolutionary process will terminate. It will cease to continue because the system conferring no function will not be preserved and passed on to the next generation.
Have the fish in New York's Hudson River evolved into super mutants? A large proportion of the river's Atlantic tomcod fish have developed resistance to certain poisons, and the mass media has heralded this as a dramatic example of evolution in action. However, far from supporting microbes to man evolution, these mutant fish have actually devolved, not evolved. That's because the fish have become resistant through a loss of genetic information. Non-resistant fish have special proteins in their cells that allow the poisons to bind. However, due to a genetic mutation, the proteins of resistant fish cannot bind the poisons as readily, so corrupted proteins have made the fish resistant. And in the poison-rich environment of the Hudson River, it's no wonder that the mutated gene facilitating resistance has quickly spread throughout the tomcod population. It is misleading to call these changes evolution, because evolution requires the addition of new genetic information, but these resistant fish have only demonstrated information loss.
To find out more from Creation Ministries International, visit our website, creation.com.
Did you know that part of the geological column is found upside down in Pakistan? At the foot of the Karakoram Mountains in the Salt Range Formation, scientists have discovered fossilized plants and insects. From an evolutionary perspective, these remains belong to the upper part of the geological column. However, this formation lies beneath Cambrian rocks, which are supposedly over 400 million years older. So why are the fossils of more complex life buried below fossils considered much more primitive? This is a vexing evolutionary problem. Discoveries such as these support the biblical version of Earth history, whereby the geological column is primarily a consequence of the global flood and its aftermath. It is no surprise that there is some order in the fossil record, such as sea creatures buried beneath land creatures and mobile creatures such as birds near the top. But, as the Salt Range Formation testifies, the ordered charts that adorn textbooks aren't necessarily what we find in the field.
To find out more from Creation Ministries International, visit our website, creation.com.
Now, the people that published this paper were very shocked at their findings, and there was a huge outcry from the evolutionary community when this was published. because it isn't right, right? According to their story, it's a major problem. So the people that published the paper said, using our empirical data to calibrate the mitochondrial DNA molecular clock would result in an age of mitochondrial Eve of only 6,500 years ago, clearly incompatible with the known age of modern humans. Like, it can't be right, right? Because it doesn't match our evolutionary time frame, so we're just going to ignore it. And they come up with all kinds of rescuing devices, right, to try to make it work. But the fact is staring them in the face that it's a major, major problem when you don't assume that human-chimp ancestry. You don't get the date that they would expect. Now, it's really interesting because Dr. Nathaniel Jensen, who is a geneticist here at Answers in Genesis, he's been looking at mitochondrial DNA sequences. They're in databases. You can compare them from humans all over the world. And what he found is that what we observe with the mitochondrial DNA is absolutely consistent with the biblical time frame and absolutely inconsistent with an evolutionary one. So here's what he found. He said, okay, so if she's 157,000 years old, like the evolutionists say, at least their most recent paper on this, if we use that figure, on average, we know the mitochondrial mutation rate. So on average, there should be about 1,000 differences that we find in mitochondrial DNA, okay, among humans, about 1,000 differences. If she's only 6,000 years old, like the biblical time frame, we should on average only see about 20 to 79 differences because it's less time, right? So less differences. And so he looked at that because he looked at human mitochondrial DNA sequences and databases from people all over the world. And lo and behold, what you observe is on average there are 77 differences. What is that consistent with? Absolutely consistent with the biblical time frame, and by orders of magnitude inconsistent with an evolutionary one. He's even shown this for several animals as well, the exact same thing. There's no way, when you look at the DNA, it is so stinking similar. There's just not that many differences. It doesn't even matter what people group you are. We just don't see that many differences. And that's exactly what we would expect within the biblical time frame that we have of only 6,000 years. So in summary, the male and female ancestor of us all, the biblical Adam and Eve, we know they lived very recently. We know that from the biblical genealogies and chronologies. We know that was about 6,000 years ago. Mitochondrial DNA analysis is absolutely consistent with that. So evolutionists have a lot of explaining to do with this because it just doesn't match their ideas of the time frame. And even if you look at different people groups, now notice I don't use the term races because there are not multiple races of people. We all come from Adam and Eve, so we are the human race. There are different ethnic groups, okay, different people groups, but we all kind of diverged out. Those kind of started after the Tower of Babel. So it's only been about 4,000 years ago or so since that. So of course there's just not that many differences because it's just not that much time has passed.